All right. This semester, we worked through the Science Education Alliance, more specifically the program Seed Genes, to perform molecular and phenotypic characterizations of the mycobacteriophage Brusacoram. To begin with a little bit of background information on the large scale end of the spectrum, as a program, we are working to determine how phages can positively impact human health. With many bacterial infections becoming resistant to common antibiotics, which usually target metabolic or biochemical processes, we are investigating ways in which phages can aid in combating those issues. It has been found that phages can use a high level of specificity to kill bacteria, including the antibiotic resistant, by destroying the cell walls and membrane without ever becoming resistant themselves. Now emerging as a process known as phage therapy, novel phages are being used to treat numerous multi-drug resistant infections. Because phages are still relatively new to the scene and a better understanding of them as well as their gene functions are needed, that's where we as undergraduate researchers come into play. So now moving to the experimental overview, one phage of interest this semester for us was Brusacoram, of which an electron micrograph image can be seen at the top left corner here. And we looked at each of its genes to determine function, toxicity, and ability to confer. In determining the function of each gene, we began by looking at the Famarator genome map, which is pictured below the title here, and the phages DV gene list. With the Famarator genome map, the various colors at the top here indicate nucleotide sequence similarities. So for example, the purple found over here, and even over here indicate the most similarities, while areas that are in red, such as here, here, and over here, might indicate very little similarity. Other colors are assigned at random, but there's really no key given as to what they mean. Some of the genes we did notice on the Famarator genome map had no known functions compared to others which had known functions. Next, in order to determine the toxicity and ability to confer, we followed a variety of protocols to prepare the Brusacoram genes for those steps. First, we began by amplifying the DNA of the individual genes through PCR. Then we performed gel electrophoresis to separate the DNA fragments according to their base pairs. Following that, we column purified the samples to clean the DNA and continued with gel extraction to further separate genes that needed to be better cleaned. Shortly thereafter, we completed an isothermal assembly reaction to construct a plasmid from backbone DNA with the inserted Brusacoram genes and generate viable gene clones. With the successful results from the isothermal assembly, we then moved on to doing transformation of bacteria. Using M. smegmatis, we introduced the cloned Brusacoram genes to the bacterial cells and generated large amounts of plasmid DNA, which were plated and used to perform the cytotoxicity as well as defense assays. That is as far as we got in the lab in regards to testing and running protocols. However, the next steps, which can be seen at the bottom of the experimental overview, would be to clone the Brusacoram genes into an E. coli expression vector, perform a protein-protein interaction assay, and do sequence candidates of the genes. Then over here in the center of the poster, we have the bulk of our experimental work. Starting with the cytotoxicity assays, we worked to determine if the tested Brusacoram genes were toxic to the bacterial host M. smegmatis. Using plates containing the antibiotic kanamycin in either 10 nanograms per microliter, 100 nanograms per microliter, or none of the inducer molecule ATC, which induces expression of a gene, we spotted serial dilutions of Brusacoram genes from 3 different plasmid DNA samples, as well as positive and negative controls. And at the top of this panel here, moving from left to right, we can see the plates with the no ATC, the 10 ATC, and the 100 ATC. And you can also see the sequential serial dilutions going from top to bottom here. Then after the plates were spotted, we let them sit and incubate for a total of 5 days at 37 degrees Celsius and assess the growth. Again, at this top image here, we can see that there has been good plaque formation across the dilution factors, and it can also be noted that there is a pink color in some of the plaques as well, such as over here on this far right plate. Being that expression of non-toxic genes results in normal plaque growth, and expression of toxic genes results in no plaque growth, we could deduce that this gene 19 was non-toxic to the M. smegmatis host. We could also deduce that gene 19 was successfully expressed due to the faint pink color. Moving down to this other image, using the same antibiotic and ATC containing plates, as well as method of serial dilutions, it can be seen that Brusacoram gene 46 has vastly different results after 5 days of growth at 37 degrees Celsius. With significantly less plaque growth on the plates containing the 10 ATC and the 100 ATC, we could conclude that gene 46 was toxic to the M. smegmatis host. However, there was a prominent pink color in the second plate here and third over here in the plaques that did grow, so we could conclude that expression occurred. Then moving on to the defense assays on the right here, we worked to determine whether the expression of Brusacoram genes would protect the bacterial host, M. smegmatis, from infection by other phages. Using plates containing bacterial lawns of kanamycin and either 100 nanograms per microliter of ATC or no ATC, we spotted dilutions of Brusacoram genes and controls. After allowing the plates to incubate for 3 days at 37 degrees Celsius, plaque growth was observed. In the figure here, it can be seen that both Brusacoram gene 66 and 28 had clear countable plaque formations to some degree, as did the controls, which are right here on the right. Of those formations, we took the dilution factor with the most countable plaques and performed titer calculations for the Brusacoram genes as well as controls, which determined the concentration. With those calculations, we moved on to find the efficiency of plating, which is known as the EOP, which is also a ratio of the phage titer obtained versus the titer of the control. Seen in the tables to the right, far right over here, we can see the titer calculations as well as the EOP calculations for both genes 66 and 28. So an EOP of anything less than 0.1 would confer its defensibility, while anything larger than 0.1 would confer no defense. And it can be seen here in the tables that neither gene 66 or 28 showed the ability to confer defense. The rest of the Brusacoram genes that we tested in the lab had the same results, which was not shocking, as very few genes have been shown to have defense with this assay type. Moving on to the discussion in further directions. So in conclusion, we worked with all the Brusacoram genes and found that 72 of 78 were successfully amplified by PCR, and 49 genes were successfully cloned into an egg smegmatis expression vector. With the cytotoxicity and defense assay results, we were able to find that 18 of the genes were toxic, including some that had no known functions, and none of the genes were identified to have conferred defense. Going forward, more work will be done to figure out the function of the unknown genes and find methods of conferring defense better.